SARS-CoV-2 is a highly contagious virus responsible for the notorious respiratory illness COVID-19, which rapidly spread across the world in late 2019 and early 2020, unleashing a devastating pandemic that persists to this day. Vaccines were introduced and recommended to the Australian population in February of 2021 to combat the virus. Since then, new variants of the virus, such as Omicron, have developed. Variants arise when a virus has many opportunities to replicate throughout a large population. During replication, the genome sequence of the virus may evolve to become stronger, better transmitting, and more resistant to vaccine performance. So, just as the virus develops, vaccinations have had to develop too. The bivalent mRNA vaccines, Pfizer and Moderna, target different variants of the virus all at once to offer broader protection. They utilise a nucleoside-modified mRNA that encodes the viral spike glycoprotein found on the surface of the virus. Our cells are given instructions by the vaccine to make this S protein and display it on the cell surface, encouraging the body to make antibodies.
But wait, does that mean the vaccine gives us COVID-19?
No, mRNA vaccines do not contain any live virus particles, making them incapable of triggering an infection. They don't interfere with our DNA as they don't enter the nucleus of our cells.
That's great. How many times should I visit my doctor to get vaccinated?
Well, that depends on your age and your risk factors. Children aged 6 months to 4 years are not recommended to get a vaccine unless they have severe risk factors for the disease. Every person 5 years or older is recommended to receive 2 doses of a vaccine. A third dose is recommended to all people aged 6 months or older who are severely immunocompromised, who are already receiving the 2-dose primary course.
Good news, I received my 2 doses of the vaccine. I know that my body has now been introduced to the virus, so how do the cells in my body use this vaccine to protect me from the virus in the future?
Well, remember that S protein on the surface of the virus? When the vaccine is administered intramuscularly, that S protein attaches to our cells and inserts its mRNA into the cytoplasm. Our ribosomes synthesize more viral S proteins, which move into the cellular membrane bound to MHC class 1 or 2 molecules. MHC class 2 molecules are activated by the S protein and attract other important cells of the immune system. These include CD4 positive T helper cells, which have a membrane protein that binds to the S protein. Strongly activated T helper cells begin to produce cytokines that activate and cause B cells to differentiate into antibody-secreting plasma cells. The ACE2 receptor is a cellular doorway for the entry of the virus into our cells, and the S protein is the key. Viral entry is prevented by these antibodies, which can neutralise the S protein, inhibiting its binding to ACE2 in epithelial cells. CD8 positive cytotoxic T cells can also rapidly reactivate during future infections when they recognise an antigen bound to MHC class 1. Alternatively, the antibody-secreting plasma cells can become memory B cells, which respond more efficiently in subsequent infections. They can be detected in our plasma as early as 2 days after vaccination and provide long-lasting protection against COVID-19.
What kinds of adjuvants are in the COVID-19 mRNA vaccine that make it even stronger?
The lipid nanoparticle casing that surrounds the mRNA is what allows for elevated immunological response. These LNPs act as delivery vehicles, but can also damage or kill host cells at the injection site. They form particles that mimic a regular virus to ensure the safe delivery of the vaccine mRNA to the cytosol of the target cell, whilst also protecting it from nucleases. Allowing entry into the cell isn't the only adjuvant property of LNPs. They've been shown to induce IL-6 production, which is involved in optimal immunogenicity and reactogenicity.
It seems like a lot of research has been done to ensure vaccines act as optimally as possible. Is there an easier or better way to protect the population of the world from COVID-19?
Vaccine programs in general provide enormous public health value. Vaccinations save the lives of 2 to 3 million people per year. We asked some other experts to expand on this. Dr. Stephen Lee describes the importance of vaccination programs as critical to the decreased mortality and morbidity of COVID-19 patients. The vaccine protected those who are immunocompromised and subsequently decreased the overall cost to the healthcare system, especially here in Australia. In addition, he expressed that the development of herd immunity is a positive consequence of the COVID-19 vaccine rollout.
Hold on, he mentioned herd immunity. I've heard of it before, but can you give me a quick refresher?
Of course. It refers to a large proportion of the human population becoming immunised to a particular antigen, resulting in the protection of those who are immunocompromised and have never encountered the microbe before, decreasing spread and prevalence throughout a population. Despite the overall success of the mRNA vaccines, there are some issues surrounding them. Health professionals identify that there is waning immunity within the population and new variants decreasing the efficacy of the vaccine. Public apathy in regard to the virus and a lack of pressure to maintain a vaccine schedule could also be detrimental factors that influence a spike in COVID-19 cases and an increase in pressure on the healthcare system like it once had at the beginning of the pandemic. As of the 13th of March 2023, 72.3% of the global population has received at least 1 dose of a COVID-19 vaccine. The pandemic is still prevalent in society and continues to affect businesses, the healthcare system, relationships, and the personal health of many people.
The mRNA vaccine allowed the world to return to some normality and continues to protect the population every day.